The syndromic individual gene autisms is very clear that they result from spontaneous mutations on single genes that have critical functions in brain development. The most prevalent form of autism spectrum disorder, those arise perhaps from several, perhaps hundreds or thousands of different genes that produce a genetic susceptibility that then is primed to lead to atypical brain development when there are environmental exposures that are still in the very early stages of research. Exposure to air pollutants like the diesel, smoke, or the current awareness of the forever plastics are good examples of environmental exposures that we don't fully understand how they interact with these hundreds, perhaps thousands of genes. The best example of an exposure, environmental exposure leading to intellectual disability is lead in the drinking water. That's a very severe case of a very single toxic in the environment. But as we are becoming aware, there are perhaps hundreds of environmental toxins that could be interacting with many different genes to lead to atypical brain development that results in intellectual disability and autism spectrum disorder. The biggest challenge for autism research is that the most prevalent form of autism is caused by many different genes and their interactions with the environment during development. That's the reason we start studying what they're called syndromic autism spectrum disorders caused by single genes, because those can be mimicked in experimental animals. The challenge is to build that knowledge from the single gene syndromic autisms to gather a better understanding of the most prevalent form, that it is a much more complex interaction of multiple genes and the environment during development. Yeah, those are subunits of glutamate receptors, glutamate is the main excitatory neurotransmitter, chemical messenger in the brain. And they're absolutely critical for the functioning of glutamate chemical synapses. As such, they're important for every single brain function at every single synapse. So they are part of an intricate, complex molecular machinery, and therefore they could be involved in the atypical brain development. We don't work specifically on those, but we work on molecules that modulate those glutamate excitatory synapses, like the brain-derived neurotrophic factor that I mentioned earlier. The main hopeful message coming from the labs like mine in autism spectrum disorder research is that the brain of these individuals is still plastic, and even though the symptoms develop during early childhood, the brain has an intrinsic capability of malleability to fix itself. We've seen several examples of this type of intellectual disabilities, neurodevelopmental disorders with autism, in experimental research animals where the symptoms can be completely improved in adulthood. So the old dogma that you're born with this brain and you're gonna perish with this brain is being shattered from lab research work that demonstrate that we can reverse the course even when the symptoms are presented. So the message is one of hope that work from the research labs can lead to treatment even in adult individuals with autism spectrum disorder.